evening, all. Am I audible?
Good evening, ma'am.
Yes. Good evening. So
You are
I
audible, ma'am.
thank you. So I'll share my screen and we'll start with our session. I hope I'm clearly audible. Are my slides visible? Okay. I hope they are clear as well. I think just okay. I'll change my network connectivity. I think it's better now. Okay, so we'll start with our week 7 problem solving session. So, the topic today we will be covering are analysis of CNVs and SVs, introduction to RNA sequencing, RNA-seq data processing pipeline, transcriptome assembly and quantification, transcript abundance and quantification. So this course, Next Generation Sequencing Technologies, Data Analysis and Application, it is being taken by Professor Riddhiman Dhar. He is professor in IIT Kharagpur in the Department of Biotechnology. So I'll briefly introduce myself as well. I am Pooja Yadav and I am a PhD scholar and a PMRF fellow in IIT Bombay. And I am in Department of Biosciences and Bioengineering. I work in molecular virology lab. So we'll start with our session now. Okay, so till now, for the genome data analysis, till the last week session, we have done the genome analysis and we have done the variant calling by various tool, and we got to know about the particular mutations, the specific mutation that has been incorporated in our um, genome of interest. Now we want to find for today's class in like these lecture video what all has been covered are, now we want to focus on the structural variants that has been incorporated in the genome that we are analyzing. Then we'll go on the transcriptome analysis. We'll go into some RNA sequencing, how it is being done, and what are the various tools that are being used to analyze the RNA-seq data. So we'll be covering those two today. So this is the basic pipeline for RNA-seq data analysis. So after the sequencing, we here as well we are getting the raw reads. Then we'll have to do some quality control on our raw reads, which are being done by these tool like FastQC, FastX. Then we will again, as we have done for the whole genome sequencing, same thing would be happen here. We'll do mapping and assembly. These are some tools that are being used to do these mapping and assembly, and then we will quantify our reads. So in RNA-seq, why do we do RNA sequencing? We generally do RNA sequencing because we want to differentiate between the expression of our gene. So, like in one condition, our gene is getting expressed more. In case when we compare to other condition, if we compare a control with a disease condition, let's suppose a gene is suppressed in a disease condition and it is not working properly. So we analyze this RNA-seq data, and we quantify the raw reads that we are getting from both the sample. Then we can compare each other by quantifying the reads. uh, then we can say, like, we can come up with something that here this gene is what is affecting the disease condition or the normal condition. So again, these are some of the software that are used for the quantification. So so for today's lecture, the question would be based on mainly the structural variant of the genome as well as the RNA sequencing data analysis. So for today's session, so again we will have questions, and I would like you to try answering the question first, and then we will look for the concepts of these questions. So by this, our session will be more interactive. So we'll start with our question number first. Question number first, it states that identify the true statements about copy number variation, CNVs. Um, Please go through this question and try to answer this question.
Ma'am, your screen is shared, right?
My screen is shared. Are you guys not able to see it?
Uh, Ma'am, I am not able to see it. Let me rejoin again.
Okay. Can anyone else confirm whether you guys are able to see the screen or not? See my slides. Okay. Okay. Is it visible now? Deepak, it was it right?
Yes, ma'am, it's visible now.
Okay. Okay. So please go through this question and try to answer this question. So copy number variation are basically there are different in gene copy number. So how do we get difference in a gene copy number? So based on this, try to answer this question. We have one answer, which is all are right. We have another answer, A, C, and D. The correct answer is A, C, and D. A, CNV is a part of structural variation that involves gain or a loss of a specific DNA fragment. So copy number variation is again a structural variation where there is a difference in the genome sequence of the data, which is structural variation. It either involves gain or a loss of a specific DNA fragment. Due to gain or a loss of specific DNA fragment, we tend to have a change in copy number of our gene. If there is a loss, then there is less gene or no gene at the specific site, or if there is a gain or a duplication happen of a particular gene, then we have a higher copy number of a gene. So the B option, it states that CNV occur where there are change in the order of DNA segments in the genome. So there, for the copy number variation, we are quantifying the gene. We are not saying whether the segment has changed its orientation. These are other structural variant. They are not involved in the copy number variation. Copy number variation simply means there is difference in the number of genes when we quantify it. For a different sect of, if we are comparing a genome from sect 1 and from the sect 2, there is a difference in the gene quantification, then we can say that there is a copy number variation. Order of a DNA segment, it is a structural variation, but it is not a copy number variation. C, it states CNV can occur both at coding or a non-coding region in the genome, which is true. A duplication or a deletion can happen anywhere in the genome, whether it is coding or a non-coding region. The D stands for deviation in read depth from the expected copy number can indicate the presence of CNV. When we quantify our raw reads, when we quantify our read data, if there is a duplication of a gene in a sect 1, then we will get a higher depth or a higher read count in that particular sect when we compare to the other sect. So the deviation in the read depth from the expected copy number, it can indicate the presence of CNV. So which is also correct. So I'll share this link with you guys so you can go through it for a while, so you can get a clear idea about what are copy number variations. Just go through this link for a minute. Just go through this link. So here you can see if this is a reference genome and this is our test sequence where we want to analyze our data. So these are the genes which are annotated by different shapes and color, and here is the duplication of these particular gene has happened. And if we compare, there are multiple reads where the duplication has happened. When we compare to, there is increase in the read numbers of where the duplication has happened. So just go through the link, just read the manual. So the correct answer for this question would be A, C and D. I hope this question is clear. Okay, we'll move to our next question. Question number 2. Identify the true statements from the following. A. Insertion, inversion and translocation are different types of non-CNV structural variants. B. CNVs are not part of structural variance, the non-CNV and non-CNV SVs can lead to the formation of multi-allelic CNVs. Non-CNV structural variation involve change in the structure of DNA but do not involve change in copy number. Please try to answer this question. We have one answer A, D. Are there another answer which might be correct apart from A and D? A and D are correct options. So A states insertion, inversion and translocation. They are different types of non-CNV structural variation, which is true. So when there is insertion or inversion or translocation, uh, they result in a genome different genome orientation when we compare to our reference genome, which is a part of non copy number structural variants. CNVs are not part of structural variants, which is not correct. CNVs, they are part of structural variants. C. Non-CNV SVs can lead to the formation of multi-allelic CNVs. So, non-CNV CVs, they do not lead to the formation of multi-allelic CNVs. Only CNVs, they, because there is a duplication of a particular gene which leads to the multi-allelic CNVs, not the non-CNVs. Non-CNV structural variation involve change in the structure of a DNA, which is true, but do not involve change in copy number. Again, it is also true. Uh, so just go through this link. Try here. Different structural variants are given and how they are formed is given. So here are both CNVs as well as non-CNVs structural variants. So CNVs, they basically happens with deletion and duplication of a particular gene, which leads to the change in the copy number of particular gene. While insertion, inversion and translocation, they are non CNVs structural variants which change the genome orientation of the genome, not the copy number change of a particular gene. And there are other complex variants as well, where multiple inversion or inversion duplication or deletion inversion happens. So they are the combination of these independent SVs. So the correct option for this question is A and D. I I hope this question is clear. We'll move to our next question, question number 3. So here a reference sequence is given as well as a sample sequence is given. In this question, what is asked, what kind of structural variation is this? Please go through this question and try to answer this question. So there is two combination of two SVs we can see. Okay, so we have one option which is B. Is there any other option apart from B that might be correct? For this question, B option is correct. So here the combination of two SVs we can see. So you can see this ABC, this region has been inverted or inversion has been happened, and we can see that, so inversion of ABCD, you can see the entire the segment has been inverted, and there is also the duplication of A and D, which is of the same orientation that of the original. So a paired duplication and inversion has happened in this SV. So the correct option for this question would be B. So this is the example of complex structural variation. So where the combination of a different SVs which results into such complex SVs. So I hope this question is clear. So we'll move to our next question. The question number 4, it states which of the tools used to use the Reed pair distance method for the identification of structural variance. Please go through this question and try to answer this question. So basically what here we are doing, if there has been some insertion has happened in the genome, when we compare the read length, if there was no insertion has been happened, and there are two conditions where at one insertion has happened, at two the other condition is insertion has not happened. So there would be distance in their reads, because it's a pair and sequencing, so there would be a difference in distance of these reads in the two conditions. So we are finding that difference which might indicate some structural variations. So what are those tools which are able to identify these read pair distance? So here are some options given. You need to select options from them. So we have one answer, D and E. apart from D and E, is there any other answer that might be correct? So the correct answer for this question is D and E, Beamer and break dancer. So what is basically what we are doing here? um, so a paired read distance, it includes the full read sequence. So for example, for paired end reads, the full read sequence is forming the beginning of the forward read to the beginning of the reverse read. So let's suppose this is our uh, this is our genome, the reference genome, and we have a two sets where insertion has happened. You can see. So this is our read 1, this is our read 2, and if we compare this with the reference, we can see there is a huge difference in the distance of these two. So if we find the read pair distance among these two reads, we can comment on something that yes, there might be some structural variation has happened. Here we can see if there is an increase in the read pair distance, we can see there is insertion or a duplication has happened. Or if there is decrease in the read pair distance difference, then we can say that there might be some deletion has happened. So based on such inferences, we can comment on the type of structural variation by calculating the read pair length distance. So these are the two tools, Beamer or breakdancer, which allows us, where we give our data, which allow us to find the read pair difference between among the reads. So the correct answer for this question is D and E. So I'll share the link. Okay. For these, so maybe you can go through them later to find about it more. You can go through them after the class if you are interested, so you will know how these tools work. So we'll move to our next question. Which of the methods are particularly effective for detecting small structural variants whose breakpoint is located within the repetitive or complex genomic region? Please go through this question and try to answer this question. So I'll share a link. Please go through this link and then try to answer this question. We have one option C. There can be multiple option correct for this question. Okay. Is there any other question, another option correct, including C? Please go through the manual. Okay, so we have another option A and C. Let's go to the manual. So, there are few ways where we can detect CNVs. One is reed pair, the second one is split reed depth, and assembly. Reed pair distance, we have seen that how we can calculate our reed pair distance. So, reed pair distance, what it does is it gives the distance among the difference in the reeds. So, basically, here we can find a complex structural variant, but in case of split reed, what happened, let's suppose there is a deletion has happened, or duplication has happened, or an insertion has happened. So our reeds, they will be split, are a single reed. So here in this case, our single reeds, they are not splitting, while here in this case our single reeds, they tend to split, and then they can cover. So these unmapped reeds are potential source of breakpoint at the single base pair level. So here our reeds, they are splitting. Our reed 1 and reed 2, they are splitting because they are not covering that certain portion where a breakpoint has happened. So these, they will remain unmapped, and this is a source of a breakpoint at a single base pair level. While in case of reed depth, what happens is there is a difference in the reed counts. Here there is no splitting has happened, but there is difference in the reed count, where in one case there is higher number of reeds, while in the another case there is there is less number of reeds. But it does not give us the information about where the breakpoints has happened, because here the reeds are intact. While in case of assembly based method, here also the reeds are basically, they tend to split where certain deletion has happened. So both split end and the assembly based method, they are effective for detecting small structural variants whose breakpoint is located within the repetitive or a complex genomic region. It is because here the reeds are also splitting. Reeds are not splitting, but reeds are covering the different portion of the genome, and there are certain unmapped region in both of these which tend to give us the information about the breakpoints in the in the particular locations. So the correct option for this would be C and D, because for the D there would be certain difference in the assembly, where we can see the assembly has not been accurate as of that reference genome. Then we can also comment on the breakpoints. we can identify where these differences are occurring, and we can comment on the breakpoints of that particular region. So the correct option would be split read method and assembly based method. So the link that I am sharing, after the class you can go through each of the link, where you can also, like, you can find how these, like, specifically these are being done, so you can get a clear idea of the questions that are being discussed here. So we'll move to our next question, question number 6. What are the files that are required as input in the feature count algorithm? So here we are quantifying our reads. This is one of the tools that are used to quantify the reads, which is feature count. So so what are the input files that we give to this tool? So where we can get our input, we can get the output. So please go through this question and try to answer this question. So here, so in the first step we had the raw read data. We check its quality score, then we map an assembly, and then we count, we do the quantification of our read to see if there is a structural variations that has been happened. So we have discussed what sort of structural variation that can affect the quantification of the reads. So this is one of the tools that is used, feature count. So we need to give some files where it can detect what are the read difference. So what are those files that we give to that tool? So in this question, some of these file options are given. You need to select from these, and there can be multiple options correct for this question. So I'll share the link. Okay, we have one option B and E. Okay, so please go to this link. Please go to the link and try to answer this question again. Please go through the link for the manual I've shared and try to answer this question. Okay, so we have one option B, D, E. Okay. Okay, let's go to the link. So, okay, so feature count is a highly efficient read summarization program that counts map read for the genomic features such as genes, exons, promoter, etc. It can be used to count both RNA-seq and DNA-seq reads. So the feature counts it take input as BAM and SAM file. So after the alignment we get BAM and SAM file. So for the input it requires BAM and SAM file, and the annotation file, the gene annotation file that we got, which is our either our GFF file or simplified annotation format. So as an input it requires the alignment file as well as the gene annotation file. So the correct option would be A, C and D. So the correct option is A, C and D. So please go through this manual, take few minutes. Yes, GFF and GTF files are the same files, just there are few extra information in the GTF file is present. It is again the genome annotation file as like GFF. So I would suggest to go through all these manuals so you will get clear idea. So we'll move to our next question. Which of the process is generally responsible for the generation of mRNA isoforms? So mRNA isoforms are basically different forms of like a gene. We have a gene. We have a genome where we can see different genes are present. So how these genes are combining with each other to form an mRNA, and uh, these can combine in different form and making these isoform different isoforms. So what is the process of generation of these mRNA isoforms? So in a gene we have exon and an intron. So what are these? How are we getting mRNA isoforms from these gene combinations? Please answer this question. Okay, let's see. So we have a gene, and the gene has a coding as well as non-coding region. So the coding region is the exon region, while the non-coding region is the intron region. So when the DNA is transcribed into RNA, so it is not the RNA that from where the protein is translated. So some processing has happened to this RNA, and then it form a mature RNA, which is mRNA. So this mature RNA become can be of a different combination of these exons present in a particular gene. Let's suppose there are 5 exons that is present in a particular gene. So the mRNA, it can form including all the exons, or it can exclude one of the exon and form a different mRNA, while again it exclude one of the mRNA, one of the exons, and for one of the different exon and forms a different mRNA isoform. So it can also happen that there is only one exon that is coming, or just 2 exon, the combination of 2 exon are coming. So they are basically forming the mRNA isoform. So this isoform we are saying in comparison of that particular gene, the parent gene. So there can be a different isoform can be formed from that particular gene because of the combination of the different exons, and this process is called alternative splicing, where they so splicing is basically the intron part is getting spliced and the exons part are joining. So alternative splicing, the different combination of these exons are getting joined and forming different forms of mRNA, which leads to different protein formations. So these mRNA, they synthesize different proteins. So they are basically when we compare to each other, they are isoforms, but again they are synthesizing different proteins. So that is why we have the same gene everywhere, but in our organ, but we synthesize different protein at each organ. So let's suppose liver, we have a same gene in liver as well as heart, but the protein where in the heart is different from the protein in the liver, because the main reason is because there is alternative splicing of the gene that happens, which is forming different proteins from the same genes. So the process by which these mRNA isoforms are being formed is alternative splicing. So I hope this question is clear. We'll okay. So we'll move to our next question. Question 8. Which of the following options are the advantages of long read direct RNA sequencing? Please go through this question. So I'll share the link for direct RNA sequencing. You can go through this link. You can read about how long direct RNA sequencing is being done. Please go through the link and try to answer this question. So we have one option A, B, and D. Okay, so for a long read direct RNA sequencing, we generally do by this either Oxford Nanopore, and here this is we are not synthesizing um the complementary strands. We are directly by difference in the current signal coming from the different combination of the sequence, we are directly getting the signal and try to identify what can be uh, the sequence. So the option B, it is sequencing by synthesis process, is not correct, because here we are not synthesizing any complementary strand. We are directly sequencing from our uh, DNA or RNA here. So do remember the principle for Oxford Nanopore before answering this question. So long read either. So here it is in here. SMRT is given. Okay. So basically, long read RNA sequencing, they are basically done by Oxford Nanopore, and here the we directly detect the change in the current sequence signals when we pass our genome, or here RNA, from that nanopore. So if we pass, if it is getting the current signals, we can detect RNA modification, because if there is any epigenetic modification that has happened on the RNA, there will be a different current signal that we will be getting. It is a sequencing by synthesis process, which is not correct here, because we are using the Oxford Nanopore principle. Biases and error are less well understood, which is also not correct. Is able to identify isoform, it is correct, because again we will be getting different signal for the different combination of our gene that we have seen here as well. So from a single gene there can be a multiple isoform that can be formed, which we can detect from long read sequencing, because from here we are able to find the different signals. So the correct option would be A and D. It can detect RNA modification and is able to identify isoforms. So here is a Nature article, I will share it with you. How these epigenetic modification can be identified based on long read direct sequencing. So you can go through this paper if you like. So it's an interesting paper that I found. Just you can get an idea how these things are being done. So if you have a free time, you can go through these links. So the correct option would be A and D for this question. We'll move to our next question. Question number 9. Which of the following processes is useful for analyzing expression of two genes that overlap with each other on opposite strand? Please go through this question and try to answer this question. So if there are two, we know that there are two strands of the DNA. If there is overlapping region of two genes, either on the reverse as well as the forward strand, so uh, to analyze this sort of data, what sort of sequencing we should do or we should go for to differentiate that this gene is coming from this strand and this gene is coming from this strand, because overlapping can happen among these genes which are present on the different strands. where a false signal can come, because the read can overlap among these two different genes, and we can over quantify a read which is coming from, let's suppose, okay, I'll just okay, so I just, so let's suppose this is one strand, this is another strand, and gene A and gene B. So here gene A is present and gene B is present, and some region of gene A and gene B are overlapping on these strands. So when a read is coming, let's suppose a read is coming from this, okay, let's suppose this is the read, this is our read, it is coming from gene A in a sequence, but we can see that here this part of the read is covering the gene B, and for a gene B we can over quantify our read, and here the read quantification specifically in the transcript analysis is very important, because these expression level, they give us the information about how these genes are being differentially expressed. But if we compute this read for this gene, we can get a false information from here. So in this question, what is asked, for analyzing expression of two genes that overlap with each other on the opposite strand, what sort of sequencing should be done? So I think now this question should be self-explanatory if you go through this options. So for these kind of data, we will be doing strand specific RNA sequencing, because we cannot rely on pair and sequencing here for both the strands altogether, because here we can over quantify our read depth for one of the genes, because here genes are overlapping and they are on the different strands. So for this reason, we'll have to do the strand specific sequencing. We'll separate these strands, we'll make cDNA of each strands, and we will do the different sequencing for each of the strands. So we'll have to separate these strands first, and then we will do the strand specific sequencing. So the correct option would be option D. So I hope this question is clear. If there are doubt in this question, so you guys can ask me. So to basically not over analyzing our reads for a particular gene, and we have the information that there is an overlap of these two genes which are present on the different strands, so we will go for the strand specific RNA sequencing, where we first separate both of the strands and then sequence them separately. So if this is clear, we'll move to our next question, which states that which of the following statements about HiSAT's algorithm is correct. So I'll share a link with you. Go through this link. and try to answer this question. So we have read about Burrow-Wheeler transform and FM indexing in previous lectures. So this new algorithm, which is, it is not a new algorithm, it is again a combination of both BWT and FM indexing that we have already read. So please go through the link and try to answer this question. Okay, so we have one option C and F. Is there any other option that might be correct apart from C and F? So let's go to the link. So HiSats is a fast and sensitive alignment program. So it is again, uh, it's a package for aligning our raw reads to our reference sequence, mapping our next generation sequencing data to to our reference genome. So it is based on the extension of BWT and FM index that we have already covered what how BWT and FM indexing work during aligning our raw reads to our reference genome, and it works. It is the combination of both again BWT and FM indexing, and it is this algorithm is also a splice aware aligner since we are aligning our RNA raw reads to our reference genome. So it should be, this alignment should be splice aware, where uh, we should know what where we have a different combination of RNA and mRNA isoforms. So this algorithm, it covers this information as well. So in this case, it is a splice aware aligner. So the correct option would be C and F. So please go through each of the manual that I've shared. So if you go through these manuals, so these concepts will be better clear of. Okay, so we'll move to our next question, question number 11. Which of the statement is true about star? Please go through this question and try to answer this question. I'll again share the link of the manual for the star. Please go through the manual and try to answer this question. So again, it is an alignment tool. So how this alignment is done by this tool, we need to find the correct options. So we have one option, A and C. Are there any other options that might be correct apart from A and C? Yes, so if I go to the manual, so please go through the manual that I've been sharing, so you'll get the clear idea how these tools work. So here we have again, it's so this tool, the star, it is for the read alignment to our reference genome. It is again splice aware mapping to the genome. So this star aligner, it seed searching, it perform two process. It search for the seeds, clusters, stitching and scoring. And if we go through all the steps, so the star follow, it's a sequential seed searching, while the reads that are matched for maximum mappable prefix against the reference genome sequence. So two steps it is following. It is sequencing, searching for our sequential seeds. Then the reads are matched to the maximum mappable prefix. Then here in this algorithm, we'll not go into the algorithm, so we'll have some prefix. So these reads, they'll match to the maximum mappable prefix. Against here it will find some locations based on few algorithm, which is the maximum mappable to the reference genome. So first it would search for the seeds, where we will get the seeds, then it will map to the reference genome. So the correct option would be A and C. So please read the manual, take few more minutes, please go through the manual, you will get the clear idea how these are being done. So seed searching is happening. uh, the longest matching sequence are called maximum mappable prefix, where our seeds would align. So we'll move to our next question, question number 12. Which of the algorithms follow the alignment free method? So these are for the quantification of the reads that we are getting, and in this question what is asked, so for these quantification tool, these, how many of these quantification tools are alignment free method, which follow the alignment free method, where we do not actually align our reads to the reference and then quantify, we are directly quantifying our reads. So I'll share few links for just read these. Okay. This is for salmon, this is for ISATs, this is for sale. Please go through it and try to find how these quantification is being done by various tools. Okay, so for cholesterol not given, right? Okay. So in the pipeline, what we are doing, uh, we are quantifying our reads. We are doing the quantifications of our reads against the gene to our genome of interest with the reference genome. So in this question, we are doing this quantification uh, without the alignment of raw reads. So the correct options are these: Callisto, salmon, and sailfish. These are the tools which do not do the alignment before quantification. These are quantification alignment free tools. I have shared a link for these tools. Please go through it and try to see how these are done. I have not shared for Callisto, I guess. So we'll move to our next question. Please go through these manuals so you'll know how these are being done. Like, why do we need to do the alignment, or what happens if we do the alignment free quantification, and what are better, should we do the alignment or should we not do the alignment? So you'll get an idea how these are done. We'll move to our next question. Which of the following statement are true regarding the use of BWT, Burrow-Wheeler transform? So we have already studied about the Burrow-Wheeler transform in the previous lectures. So where we did the alignment for our whole genome sequencing. Please go through this question and try to answer this question. So if you remember correctly, so we add the dollar sign at the end of our sequence, and then we form a BWT, a Burrow-Wheeler transform of that sequence, and then we form a lexicographical, meaning we arrange those sequence alphabetically, and we give it, we term that as a first column, and we term our BWT as a last column, and then we try to map our read with the algorithm we made. So remember that algorithm and try to answer this question. The first option, it is stating BWT is used to compress the reference genome for efficient storage, which is correct option, because in BWT we are storing just the BWT and the first column and the last column of the sequence. So I'll not go through it how that last column is being made, because we have already covered it. So we add the dollar sign at the end of our sequence, then we rotate our sequence. So first we take the dollar sign at the initial position, then at the second position, third position, fourth position. That's how we get our last column. Then we sort the last column lexicographically, and we give this column as our first column. Then we store these column, and when we here we are not storing the entire data, we are storing just this BWT. So it is basically compressing it is the compression of reference genome, which is taking the less storage. So this option is correct. BWT is used to compress the reference genome for efficient storage. BWT enable rapid and memory efficient alignment of sequencing reads, which is also true, because when we do this alignment using BWT, we are taking our seeds or some k-mer of our sequence, and then we are seeing where our k-mer is exactly aligning to our reference genome by using this FM indexing. So by doing this FM indexing, it is comparatively very fast and it is very memory efficient alignment. Then this option is correct. The C option is BWT is responsible for handling alternative splicing event in the reference genome, which is not true. It does not handle the alternative splicing event. BWT is used to identify exact matches seeds between the read and reference genome, which is also true, because here we are doing the alignment, and it can identify the exact match between the read and the reference genome. So the correct option would be A, B and D. So let's go to our next question. Which of the transcript quantification algorithms use expectation maximization algorithm or a simple probabilistic model for isoform quantification? Please go to this question. So I'll share few links with you. So I'll also share about this algorithm, expectation maximization. So we'll not cover how exactly is this done. I'm sharing the reference. So if you want to know more, you can go through these reference and try to see how exactly they work. Slide not visible. Are my slides not visible to anyone? Aruna, can you please rejoin the lecture? I think the slides are visible. Can anyone else confirm whether my slides are visible or not? Okay, it's visible. So Aruna, please rejoin the lecture. So there might be some network issue from your end. Okay, so we have one option A and C. So there are different ways where we can quantify our reads. So there are different ways and different algorithms. So there are tools based on these particular algorithms. So one of the algorithm is expectation maximization. So we want to answer. So what tools use these algorithm? So the correct option is A and C. So these are the tools for transcript quantification, which uses this expectation maximization algorithm. So I've shared the link with you. You can go through this link and see how these algorithms, what are these algorithms and how they actually work. So I have shared a Nature article with you. Please, I would highly suggest to go through this article where it has been clearly explained. So how these algorithm works. So I found this article very interesting. Where, so if you have time, please go through it. So we are not using the algorithm directly, but we are using the tools which are based on these algorithms. So if we know the algorithm the tools are using, then we are able to analyze our data more efficiently. If we do When we compare, if we don't know the algorithm which our tools are based. So it's better to know these algorithm, to know the basic of our tools. So the option A and C are correct. They are using expectation maximization algorithms. So that was our last question for today's lecture. So please, I would again suggest go through all the manuals, read all the manuals, and probably most of the questions that you have will be clear if you read. There are some basic steps for both whole genome sequencing and RNA sequencing, so that we have already covered here. So data quality control, mapping, alignment, and further the analysis. Here that was for the whole genome sequencing, and this is for the RNA sequencing. These are the same. There are particular tools that we used at each steps, and there are some algorithm which these tools are based on. So just I have shared most of the manuals for most of the tools and algorithm. Please go through it and learn more about them, so you will get a clear idea what are happening here and why, like what can we do with our data. If we analyze our data, what is the better option to analyze our data. So that's all for today's lecture. If you guys have any doubt for today's lecture. So if there are no doubts, we can end our session here. We'll meet next week at the same time. So if you guys don't have any question, we can end the meeting here. You guys can leave the meeting.